All right, welcome back to the Chapter 13 lecture on meiosis. This lecture can be used in conjunction with the Chapter 13 lecture review sheets, and it can also be used with the printable versions of these slides, both of which are available on Blackboard. Now in this chapter, we're going to be discussing a new type of cell division. In Chapter 12, we talked about two other kinds of division, binary fission and mitosis. And in this chapter, we're going to discuss meiosis. Now, to understand those processes, we need to compare them, and we need to begin our comparison with the purpose of these two types of division. Mitosis is used for growth and for healing. So whenever a body needs to make identical copies of pre-existing cells, they use mitosis. For instance, if you're growing skin cells and you need to make more skin cells to either grow larger or to heal a wound, you use mitosis. This is a form of asexual reproduction. For instance, at the bottom here, you can see the nucleus of an embryo that is dividing by mitosis from 1 into 2, 2 to 4, and 4 to 16. And the nuclei, the DNA inside each of these cells, is identical to all of the other cells. They are all the same. Now, the purpose of meiosis is very different. In meiosis, we're trying to make gametes, or sex cells, eggs and sperm cells. So meiosis is an important step in sexual reproduction. And those gametes are fairly different than other types of cells, so it's going to require a different kind of division. Next, let's discuss the different kinds of cells that are made by these processes, and hopefully that will help you understand how they are made. So first of all, we have somatic cells. Somatic cells are body cells. So these are things like skin and heart and brain cells. Now these types of cells are genetically identical to one another within an individual. So if I were to look at the DNA inside one of your skin cells, I would find that it is exactly the same as the DNA inside one of your heart cells. Now these cells are diploid. That means they have 2 copies of every single chromosome. And in humans, that means they have 23 pairs, or 46 chromosomes total. Here we have a simplified cell that only has 3 pairs of chromosomes. Now, one of the chromosomes from each of these pairs would have come from mom, it's maternal. The other would have come from dad, or is paternal. Now let's compare that to a gamete. Gametes are produced by meiosis. Gametes are genetically distinct or different from one another, and they are haploid. So what this means is that there are no 2 egg cells or sperm cells that are genetically the same. They are all different from each other. And if we could see the chromosomes inside, we'd see that they only contain 1 copy of each chromosome. They are haploid. Now in this example, the haploid number for this cell would be 3, because there's only 3 here. In the human system, the haploid, or N, number is equal to 23, because a diploid cell has 46. So half of that would be 23. Now next we need to discuss how this is accomplished, how these different types of cells are made using these different types of division. So we're going to begin with mitosis. Mitosis begins with a diploid cell that undergoes a single division, and it ends up making 2 daughter cells that are identical. So everything stays the same in mitosis except the number of cells. Here we have a simplified cell that we can look at. This cell only has 1 pair of chromosomes instead of the 23 we would find inside a human cell. Now in this particular case, this little cell might be in, let's say that, it's a terrible G, but the G1 phase, because you notice that these chromosomes are unduplicated. However, in the S phase, the chromosomes are going to become duplicated. So they're going to be copied. And so now we have 2 duplicated chromosomes that look like X's. During mitosis, the chromosomes are going to line up in the middle of the cell, and then it's actually the chromatids that separate from one another. So the X's break in half, and these identical copies go opposite ways. What we end up with are two cells, called daughter cells, that are identical to each other, and you notice they are also identical to the parent cell that we started with. So everything stays the same in terms of the number of the chromosomes. The only thing that changes is the number of cells. Now remember that the purpose of meiosis is to make gametes, to make these haploid daughter cells. So the process of meiosis is going to be pretty different. It's going to start out the same though. Here in G1, we're going to have one pair of chromosomes. So this little cell is diploid. And remember that since we're making sex cells, this little cell will either be located inside the testes or inside the ovary. But they are diploid starting out. Now those chromosomes are going to get copied during the S phase, so they become duplicated and look like X's. And then they undergo these divisions. Meiosis actually involves two divisions. During the first division, the homologous chromosomes separate from one another. So one X goes one way, one X goes the other way. In Meiosis II, the second division, the chromatids within each cell separate from one another. So what we end up with are 4 daughter cells that are haploid. Now, why are they haploid? Well, let's think about this. If we start out in G1 with 46 chromosomes, and those chromosomes get copied, we are going to have 92 chromosomes before division begins. If we divide those in two through one division, that number is going to come down to 46. And then if we divide them again, it's going to come down to 23. 46 divided by 2 is 23. So now since there's only one copy of each chromosome in the cell, we say that that cell is haploid, and its number is 23. Now another interesting thing to note with meiosis is that those daughter cells are actually genetically different or distinct from one another. Okay. First, look at the chromosomes inside the parent cell here. You notice that there's 2 of them, one red and one blue, because one came from mom, we'll say, and one came from dad, maternal and paternal. In the daughter cells, though, we only have one copy of those chromosomes. And you'll notice that the colors on some of these are mixed up, represent mixtures of the 2 sources. So they're different genetic combinations. No 2 of these cells are genetically the same as one another, and they're definitely not the same as the parent cell because they have half the amount of DNA and they represent different combinations of those genes. So they are genetically distinct. Now, before we proceed, I want you to remember that the point of meiosis is to take one diploid cell and divide it into 4 haploid cells, and that these cells are going to be different from one another. This means that each haploid cell has one complete set of chromosomes instead of 2. And this represents half the DNA necessary to make a person. Now, why do we care about that? Well, remember, what we're making here are sperm cells or egg cells. So those cells, ideally, will fertilize one another, which means that the embryo that's made, the fertilized egg that's made, will have 46 chromosomes. So it's important that they only have 23 each. We can relate this back to an analogy I gave you in the last chapter. In the last chapter, I told you that having a diploid cell is kind of like having 2 sets of encyclopedias inside that cell. You got one set of encyclopedias from one parent and another from the other parent. Now, they all have 23 volumes in them, 23 chromosomes each, and they address the same topics, but they might address those topics a little bit differently. you're not going to find exactly the same information in one book about a topic as you'll find in another book about that topic. So they both carry the same sets of genes, but there can be little variations in those genes. On the other hand, a haploid cell, like an egg cell, is only going to have one set of encyclopedias inside. So 23 chromosomes instead of 46. And the volumes within that encyclopedia represent a mixture of the source material. So in this encyclopedia set, we're going to have some volumes that came from Dad, that Dad gave you, and some volumes that Mom gave you. A combination of that information. Next, we're going to go inside those two divisions of meiosis, and I really want you to understand what type of DNA is dividing during each division. I also want you to understand that these two divisions result in the number of cells made through meiosis. So let's get into that. First of all, meiosis involves two divisions. The first division is called Meiosis I, and the second division is called Meiosis II. During Meiosis I, what's separated are the homologous chromosomes. So the DNA from each parent is separated. During Meiosis II, it's actually the sister chromatids that are separated. Again, we're going to start with one diploid cell in a testy or an ovary. And at the end of the process, we're going to end up with 4 cells that are haploid. Let's take a closer look. All right, so here we have an example cell. This is a simplified cell that only has one pair of homologous chromosomes in it instead of 23 pairs. Now these chromosomes are duplicated, so they look like X's and they're held together in the middle with a centromere. So there's that big old centromere. Each half represents a sister chromatid. So the sister chromatids within each chromosome are identical copies of one another. Now let's see what happens during Meiosis I. Here's the end of Meiosis I. What's separated? Well, obviously, one cell divided into 2, but what's separated in terms of the DNA? Let's take another look at that. Again, here's the cell at the beginning of Meiosis I. Here are the cells at the end of Meiosis I. What's separated? Well, let's take a look at the entire process put together, and I think that will make it obvious. At the beginning of Meiosis I, we had a cell with a pair of homologous chromosomes. So the 2n number for this particular cell is 4, because there's 4 pieces of DNA involved. At the end of Meiosis I, though, we have 2 separate cells, and each one only contains 1 duplicated chromosome. So the homologous chromosomes are what separated here. The one from Mom went one way, the one from Dad went the other way. And our 2n number, our diploid number, at the end of Meiosis I is now 2. It went in half. Now, what's separated during Meiosis II? Here are the 2 cells at the beginning, and here are the 2 cells at the end of Meiosis II. What's separated? Well, obviously, 2 cells divided into 4 cells, so each cell divided again. But what's separated in terms of the DNA? Let's look again. Again, here is the cell at the beginning of Meiosis II. Here are the daughter cells at the end of Meiosis II. What's separated? Well, here's the entire process of Meiosis II put together, and you can see it more clearly now, hopefully. We start out with this cell that has a chromosome number of 2, because each one has 2 pieces of DNA. Each one divides into 2 cells, and now each cell contains only a single chromosome. So we say that the N number is 1. There's 1 piece of DNA in there instead of 2. Now at this point we're saying N because these cells are haploid. These daughter cells each contain one copy of the chromosome, not two. So it's haploid. What's separated? Well, it's the sister chromatids that separated from one another. If you think about it, meiosis II is more similar to mitosis than meiosis I is. And the reason is that in mitosis, sister chromatids separate as well. Here's the entire process of meiosis put together into one big slide. And again, you can see that we start out with one parent cell, that is diploid. That the homologous chromosomes separate during meiosis I into two cells, each with one chromosome inside. And then those divide into separate chromatids. So we end up with 4 cells, each with one chromosome. Now remember that as long as these things are attached together, we call this entire thing a chromosome. Once they separate, we call the chromatids now become their own chromosomes. Next, we need to discuss a couple of the processes that make these cells genetically distinct from one another, or genetically different from one another. And the first one I want to discuss is something called crossing over. In crossing over, the chromosomes are going to trade some of their DNA. The copies from mom and the copies from dad are going to get together and swap some genes. And this happens physically inside the cell. So let's take a look at it. First thing that happens during crossing over is that these homologous chromosomes get together. So the ones that match from mom and from dad get together. And for the sake of this example, we're going to say that the blue ones are from dad, they're paternal, and the red ones are from mom, they are maternal. Now this process happens very, very early on during meiosis. It happens during a phase known as prophase I, and it happens before anything divides. So here we go. These chromosomes are getting together. They get really close to one another, and they form tetrads. Tetra means 4. So that means that there's 1, 2, 3, 4 pieces of DNA interacting here. So tetrads. There are these little protein strings that are going to tie them together. We call that synapsis. So they synapse during this process. And what's going to happen is that these chromosomes are going to overlap. They're going to overlap their arms, or they're going to overlap their legs, or both. Here they're overlapping these 2 legs. At the crossover point, the DNA is going to break, and the pieces of DNA are going to switch with one another. And you can see the result of that here. You can see that here this little piece of red DNA is now attached to the blue, and that the little piece of blue DNA is now attached to the big red chromosome. Now, at the end of meiosis, all of these little chromatids, technically, are going to separate into 4 separate either egg or sperm cells. And what we can see now is that these represent 4 different combinations of the source DNA. So these 4 cells are genetically different from one another. Let's look at a more specific example of this. Here we have a pair of homologous chromosomes, one from dad and one from mom. Now they are homologous, and what that means is that they carry the same genes in the same places. So here, for instance, at this locus, or location, we've got the eye color gene. It's the same place on dad's chromosome as it is on mom's. Here we have a gene for hair texture, and notice that it's in the same place on both chromosomes. Now remember that mom and dad can give you different variations of these genes, and so we've mixed it up here to make it interesting. Let's say that dad gave you a gene for brown eyes. Mom gave you a gene for blue eyes. Dad gave you a gene for curly hair, while mom gave you genes for straight hair. What's going to happen to these guys during crossing over? Well, in these examples, or this example, they've all gotten mixed up together. So here are the 4 chromosomes that result from crossing over, and here are the combinations of genes they carry. Because they crossed over, we're going to have one chromosome that still has brown eyes with curly hair, just like the original. We're going to have another one that has brown eyes, but now it's in combination with straight hair. That's a different combination to pass on. On this chromosome, we have a blue eye gene with curly hair, and here we have a blue eye gene with straight hair. If these are passed into egg cells, now we have egg cells with 4 different possible combinations of eye color genes and hair texture genes that might get passed on to the next generation. What does this mean in terms of reproduction? Well, you know that when you pass on your genes to the next generation, that you're only going to give them half, because you've got a partner that's going to give the other half. Now you have 2 sources for your DNA. You have DNA that your mom gave you and that your dad gave you. You have 2 copies of DNA, and you're only going to pass on one. Because those 2 sources get mixed all together before you pass the genes on, you really don't know which version of each gene you're going to pass to the next generation. You only know that the next generation, that egg cell or that sperm cell, is going to get one of your copies for each gene. Crossing over provides new combinations of the genes. The other factor that makes these egg cells or sperm cells genetically distinct from one another is something known as independent assortment. Independent assortment tells us that these individual chromosomes go into the egg or sperm cells independently of one another. In other words, there is nothing that determines whether it's mom's gene or dad's gene that you're going to pass on. Let's look at a couple of examples here. We've got some simplified cells. Here we have a cell that has 2 homologous pairs of chromosomes instead of 46, or 23 pairs rather. Now in this example, this cell has undergone Meiosis I, and you can see that in one cell, it got both chromosomes from dad, and in the other cell, it inherited both chromosomes from mom. However, there's nothing saying that it can't pass those chromosomes on in a different combination. Here we have the same cell, but after this round of Meiosis I, you'll notice that there's a chromosome number 1 from mom in combination with the chromosome 2 from dad. Over here, it's the opposite. We've got the chromosome 1 from dad and chromosome 2 from mom. So there's really no telling which combination of these chromosomes are going to get passed on. You only know that you're going to give one copy of each to the next generation. You don't know which source it will be taken from. So that's the mechanics of meiosis, kind of how it works. Next, we need to discuss the importance of meiosis. Why do we use this really complicated system to make sex cells? Well, one of the important things that meiosis does is it maintains the chromosome number. In other words, it keeps the chromosome number the same from one generation to the next. Let me explain kind of how that works. So let's say that we start with a diploid cell. It has already been duplicated. So at that point, there's 92 pieces of DNA floating around in there. If we divide that by 2 through Meiosis I, we get cells with 46 chromosomes. And if we divide it again through Meiosis II, we get cells with 23 chromosomes inside, haploid cells. Now, assuming that the egg and sperm get together, we're going to have fertilization. And so 23 plus 23 is going to give you a fertilized egg or a zygote with 46 chromosomes. So what does this mean? Well, it means that if the somatic cells of the parent had 46 chromosomes, then the somatic cells of the offspring will also have 46 chromosomes. That's important because if we didn't have that, we would have a much more complex system for controlling our genes, and there would be all kinds of weird side effects that would be hard to manage. Let's think about what would happen if we used maybe mitosis instead of meiosis for making gametes. If in mitosis we started out with a diploid cell with 46 chromosomes, we would end up with a gamete with 46 chromosomes, twice as much. If that was then fertilized by another gamete that had 46 chromosomes, well then the zygote, the egg, fertilized egg, would have 92 chromosomes, twice as many. If they also used mitosis to produce gametes, they would have gametes with 92 chromosomes inside. And then that would get together with either one with 46 or another one with 92. And so the next generation's bodies would have even more chromosomes. So by taking the chromosome number down by half through meiosis, we ensure that each generation of human beings is going to have somatic cells with 46 chromosomes. And that makes it much easier to manage. Now, the other reason that meiosis is important is because it maintains genetic variation, or increases genetic variation. When we have offspring, we recombine or we mix our genes with the genes of another individual so that our children are genetically different than we are. And all the children are different combinations of our genes. We call that process recombination, and it turns out that it's actually really important for the survival of the species. To understand this, let's consider the opposite sort of situation, something called a monoculture. Monocultures are populations in which there's very little genetic variation. In fact, in some, all of the individuals might be identical to one another, all clones. You see this in agriculture a lot, because if a farmer finds a plant that makes a nice product, he's going to want a whole giant field of those plants, because he can make a lot of that really good product. Now, having a field of clones is fine, unless a disease comes along or unless the environment changes very drastically. Let's say a disease comes along and hits this field. If all of the individuals are genetically the same and none of them have a resistance to that disease, it could wipe out the entire field. It could potentially wipe out the entire species. So there's no genetic advantage there. If, however, he had a field with many, many different kinds of plants in it, different variations, then if a disease comes along, there might be one or two that have some kind of adaptation that allows them to survive it. They can then pass those genes on to the next generation, and the next generation will be able to resist that disease much better than the last one. That allows the species to continue. If you're interested in monocultures, you might want to look up a little information on the Big Jim banana. Those big old yellow bananas that you buy at the grocery store are produced by monocultures. And scientists are a little bit worried about them because a blight could come along and wipe out the banana as we know it. It's an interesting topic. So genetic variation is important. You'll notice that within a family, no two siblings are exactly the same, unless they're identical twins, and that each generation is different genetically from one another. And I want to point out that that kind of recombination is good. Recombination produces variety, and genetically speaking, variety is a very good thing. All right, so that's the process of meiosis as it compares to mitosis. The next thing we need to do is compare the stages of these two processes. So you remember that mitosis just had a single division And we broke it down into stages like prophase, metaphase, anaphase, and telophase. Well, meiosis has two divisions. It has Meiosis I and Meiosis II. We can break each of those down into prophase, metaphase, anaphase, and telophase. And no, we do not talk about prometaphase in this process. Meiosis II has the same: prophase, metaphase, anaphase, and telophase. Now, the mechanics of what actually happens in these stages of meiosis is very similar to what we see in mitosis. It uses spindle fibers and centrioles, and it attaches to the DNA and moves it in a very similar pattern. And because there's two phases to meiosis, 1 and 2, the phase names within each half are named with 1s and 2s. So there's a prophase 1 and a prophase 2, for instance. Remember that Meiosis I separates homologs, whereas Meiosis II separates chromatids. So we're using the same machinery each time, but it's separating different things. Let's briefly go through these stages. First of all, we have Meiosis I, and the first stage is prophase 1. So in prophase 1, similar things happen as to prophase in mitosis. So in other words, the nucleus breaks down, the DNA coils up into X's, and the centrioles begin to grow the spindle fibers. One of the big things that happens in prophase 1 that's different, though, is crossing over. So this is where those homologous chromosomes get together and swap DNA. The next phase is called metaphase 1. This is where things line up in the middle of the cell and get ready to separate, and the fibers reach down and grab them. Notice here how these things are lining up. We have, instead of chromatids lining up on either side, we now have chromosomes lining up on either side of this imaginary line, because that's what's going to separate. In anaphase 1, the homologs separate from one another and are pulled to opposite ends of the cell. And in telophase 1, they reach the opposite ends of the cell, they begin to uncoil, the fibers break down, and in some types of cells, a new nucleus will form around each one. Not in all. The stages of meiosis II are pretty similar, and they're actually really similar to what happens in mitosis. In prophase 2, the DNA coils up again, the fibers regrow. In metaphase 2, they line up along an imaginary line. And notice here, if I can draw a straight line, maybe not, that they line up so that there's one chromatid on either side of the line. Okay, not a whole chromosome. In anaphase 2, the chromatids separate and move to opposite ends of the cell. And in telophase 2, it uncoils. A new nucleus now does form around each set of DNA. And then cytokinesis can begin to happen and separate them out. So those are the phases of meiosis II. Again, the machinery is very similar to what we see in mitosis, which is why I kind of sped through it. So that is the end of the material for chapter 13. What I would suggest that you do next is practice with this material. So I've given you this your turn. What I would like you to do is to make a drawing of a pair of chromosomes. One chromosome that's paternal and one that's maternal. I suggest that you use different colors to make them really distinct. And I would encourage you to take them through and draw them out as they go through meiosis so that you can see what separates and when it separates. Now I've given you this handout to help you with it, and yes, I know that it's sideways, but you can print this out if you would like, or you can just draw these circles on a piece of paper and draw through the process. I know that you can look it up in the book and just copy what you see, but that's not going to be very beneficial to you. I really want you to draw out those chromosomes at the beginning and then think about what separates in each phase of meiosis. Use the diagrams in your book and in this lecture as kind of a guide, but really think through the process, or the your turn won't make any difference to you. Right? Well, I hope you found this lecture helpful. Please bring me questions in class and we can work through them, and we'll practice a bit more with it as we get into class. Thank you very much.